going into the binding of amino acids to the transfer RNA. Remember, you have one type of enzyme, that is the amino acid transferase, that is very specific. So since it is specific and you have 20 amino acids, you know you have at least 20 types of amino acid transferases. Now we're going to talk about the translation, the stages of translation, about the initiation, the elongation, and the termination. Okay, amino acid tRNA is what transfers the amino acid to the tRNA. And again, this is specific. Now, how is it that the tRNA gets the amino acid transferred into that? So here you have your tRNA after it's folded. This is a 3D representation of that folded tRNA. And you know that at the bottom loop you have the anticodon. And you know that at accepting site here, you know where your amino acid is going to bind. Now, this acceptor stem here, for all of the tRNAs, this has the same base sequence right at the tip over there. Now, when you get, see, this is your adenine right there, the last base right there, you get your adenine. This whole tRNA is going to be recognized by the amino acid transferase, and it's going to put one specific amino acid into this last base here, which is the adenine. So here, see, at the carbon 3, at the C3' carbon, you're going to have this bond that is going to bind to your amino acid here. You don't have to know the exact structure of this, but you have to know that at that site, you're going to have the connection to your amino acid. Now, this is a covalent bond. So you can imagine, in order to make a covalent bond, you need some type of energy. So this process is not spontaneous. You're using an enzyme, and you're also using energy. It takes energy to put amino acid in there. Okay, so this is just more detail what the tRNA looks like. The important thing to know here is the acceptor stem and the anticodon. Now, as I mentioned, it takes energy to get the amino acid into that stem. So here, if you have amino acid, you're going to use energy from ATP, and you're going to activate this carboxyl group. Then you're going to get your transfer RNA and bind to that activated group. So this is the energy that you use in order to charge that tRNA. Now, amino acids bind to which part of the tRNA? They bind to the anticodon? No. Do they bind to DHU arm? Not sure. 3' end, 5' end. I do not think it's the DHU arm because I did not mention it. It's either the 3' or the 5' end. We go back to the structure and you see, oh, look at the stem over there. Acceptor stem. Which end is that? 3' end. So your amino acid will bind to the 3' end of your tRNA. Now, getting into the translation. When you get translation, you need other types of RNAs. In this case, you have ribosomal RNAs, and you have the ribosomes. But the ribosomes do not act alone. They also need other proteins that are complexed with the type of RNA. So you need the peptides and you need the ribosomes. So the ribosomes are made up of two units. They have a small subunit and you have a large subunit. Then each of the subunits will contain many types of proteins, many different types of proteins that are associated with it. And also has is composed of several molecules of this ribosomal RNA. Now, you do have different types of ribosomal RNAs if you're talking about eukaryotes or prokaryotes. For the prokaryotes, you have smaller ribosomal RNAs. So they still have two subunits. You have a large subunit and a small subunit. Now, for the prokaryotes, you have the large subunits that's about 50 S, and the small subunit is about 30. And you say the combination of the small and the large subunit for the prokaryotes, they say is about 70 S. Some here, I don't know why they chose to represent it this way. In my head, that does not add up. But just remember that prokaryotes have smaller subunits than the eukaryotes. And also, it's very important to know that these eukaryotes and prokaryotes have these different types of ribosomal RNAs that can be used for identification of some species. If you run or the codes for the ribosomal RNAs, you can identify species based on that. Again, for the eukaryotes, you have also the small subunit and the large subunit for ribosomal RNAs, but they are bigger than from the prokaryotes. So the large one is about 60S, and the small one is about 40S. When you put them together, you get the 80S combination subunit. So our ribosomes have one RNA binding site, and then 3 sites for transfer RNAs. Now, the image of how this happens is very important. Exactly the mechanism of how this happens is very important to know.
C is for blue, that's okay with me.
You're going to have to know how this works. So here we have the small subunit of your ribosomal RNAs, and this is where you're going to have the recognition site for your messenger RNA. Now remember, when you're doing the processing of the messenger RNA, first thing that happens when the mRNA is processed inside the nucleus is it gets a cap. At which end? 5' end. It gets a cap at the 5' end. There is a reason for that in the eukaryotes. That cap for the 5' end is going to help with the interaction between the messenger RNA and the small unit of ribosomal RNA. So small unit, what recognizes the messenger RNA? So here you have, once you have the mRNA that starts associating with the small unit, you recruit the large unit, and then you start with the tRNA business, which we're going to go into a lot of detail now. Here you have 3 sites at this large subunit of the ribosomal RNA here. You have an A site, a P site, and an E site. Now the way to know which site does what is just by remembering what the letters stand for. A is for acceptance or for arrival site. So A has to be something that gets there, it is arriving there. And then the P is for the peptide addition or for the processing of the chain that is being made for the amino acids. And then the E is the exit site. So 3 sites, gets in there, it arrives there, processes and exits. So P is the peptidyl site. It hosts the transfer RNA that has the growing polypeptide chain. And then the E is the exit site. It will host the tRNA that just gave up its amino acid and is ready to leave. So how do you get the translation process going on? First, you have the initiation. You have to get it started. You have several initiation factors that are bound to the small subunit of the ribosomal RNA in order to avoid it from always being connected to the large subunit of RNA. Because if you think about it in practical terms, if small and large are always together, how is the RNA going to get there? How are you going to get this whole system started? So you have to have these units that are not together just before translation starts. So now you have initiation factors that are going to help the ribosomes to get together with the messenger RNA and also the tRNA. So the first thing, you have this initiation factor 3 that is bound to the small subunit of the RNA, and it helps to keep it apart. Remember, it's keeping the subunits apart. Now, once you're ready to recruit the other RNAs to the system here, you have this initiation factor 2. Now, the IF2, the initiation factor 2, it will only recognize one tRNA.
C is for blue, that's okay with me.
So this is why methionine is right at the beginning of every protein synthesis. In order to get all of these systems together of small subunits, large subunits, and messenger RNA and tRNA together, there is only one initiation factor here that is going to put this together, and this initiation factor is bound to the tRNA that carries methionine. It recognizes the anticodon for methionine.
C is for blue, that's okay with me.
So it recognizes here the anticodon that is UAC. Now, it's important to know the AUG is the start codon. Now, how are we going to remember that? Well, we think about something ridiculous. I could never figure out how to remember that until one day I was babysitting some kids and they were watching some type of a strange movie, something like Chitty Chitty Bang Bang. And I am trying to figure out how to remember this start codon, and the thing just went auga. I'm like, oh, that's it. Is it starting? It was just light bulb, auga. That's the codon for start of the tRNA. So now you never forget the movie, and you never forget the auga signal for start of protein synthesis. And you start with the methionine. For the initiation factor IF3, that is, what is IF3 doing? Initiation factor 3 is keeping the small and large subunit apart. Now here, when you're talking about a prokaryotic system, so always talk about FMET. Remember, FMET is for prokaryotes, methionine is for eukaryotes. It might not be very important at this point, but you can imagine that since there is this difference in the methionine from prokaryotes to eukaryotes, we could use that to some type of drug development. For example, if you are going into pharmacy or into medical school or into nursing school or research, you can think about what do you do with this difference in methionine for beginning of protein synthesis. Since they are different, you know, the code is degenerate, the code is universal. So if you want to stop a particular protein synthesis and you want to develop a drug that will stop a particular protein synthesis, you could focus on little things that are different. For example, F-methionine. If you can stop synthesis of bacteria by blocking F-methionine, so you can inhibit a particular bacteria from growing and not the rest of the eukaryotic cell from processing, from making proteins. Just an idea, why is this important? Now, when you're talking about prokaryotes, you're also going to see a lot of these Shine-Delgado sequences. This is a consensus sequence. What is a consensus sequence? Is some type of code on the DNA. It's a bunch of letters that can be recognized, a bunch of bases that can be recognized, and they show up over and over as specific parts on that DNA code. And also, since the prokaryotes have a particular consensus sequence, you can imagine the eukaryotes have it as well. So in the case of the eukaryote, you have this other Cossack sequence. We're going to look into this. Okay, so here just an image of the ribosomal RNA. You have the large subunit. Which system is this, prokaryote or eukaryote? Prokaryotes, they have the smaller numbers here. 50 plus 30 equals 70 in the prokaryote RNA business. Ribosomal RNAs. So here you have the prokaryote system. When the RNA gets together there, see initiation codon is always AUG. And you have here, just before the start of the translation for the prokaryotes, you have that consensus sequence, which is called the Shine-Delgado sequence. Now, once you have the recognition for messenger RNA, see initiation factor 3 will bind to the small subunit of ribosomal RNA and get this part recognized by the messenger RNA to start to get to assembly this protein-making machine here. Now, as I mentioned, you can imagine the system is not spontaneous. You do need a lot of energy to do this in order to assemble this system. So you get your initiation factor 3. Which one is the first one that gets together there? Factor 3 gets together with a small subunit of the ribosomal RNA. So then you want to get this thing together. See here, you start to recognize that Shine-Delgado sequence. Now you are going to recognize why is it that you need to start the protein synthesis with AUG. Why is it that you cannot start with any other tRNA, any other anticodon? Because for this tRNA here, you have a specific site that will bind another initiation factor that is called the initiation factor 2. And again, this system is not spontaneous. You do need energy. The energy to get this together comes from GTP. So now you're starting to get the pieces together, you're going to call into the large subunit of the ribosome here. So now you see here you have initiation factor 3, you have initiation factor 2 that is loaded with energy. Now you have other factors that will call for the large subunit of ribosomal RNA to get together there. Once this large subunit attaches, all these other factors will just fall out of the system because the only thing you want here is the large and the small subunit and the mRNA and tRNA in this complex. So all these other factors that were helping for the system to get together, at this stage, they will just come out of the system. Now, what else happens here? To get everything stuck in one place, it's not spontaneous, you need energy. So for this process, you hydrolyze GTP, you see? You end up here with GDP and phosphate. So this is what drives the putting together of this system. See, these are all initiation factors that will help with the assembly of the small and the large subunits. Yes, you worry about initiation factor 3 and initiation factor 2. Initiation factor 3 is the one that goes together with the small ribosomal. Yes. And then the 2, IF2, is what binds to the transfer RNA, but not any transfer RNA, only the one that has anticodon for AUG, for methionine. So once that is there, IF2 comes together loaded with energy. And that energy is what's going to pull the big subunit to the system. And once you hydrolyze this energy, you get all the factors kicked out of that. Actually, you have more than these 3 factors here. You have more factors that are involved. But the most important, IF3, IF2, that comes with energy. Get the system together, and now the system is ready to start making protein. And this is at the close to the 5' end of your messenger RNA. In this case is prokaryote, so there is no cap here. Remember, there is no processing in the prokaryote system because there's no nucleus. There's nothing in there to compartmentalize messenger RNA from the rest of the cytoplasm. So there is no processed messenger RNA for the prokaryotes, yes. But once they are together, they will just start to translate the messenger RNA. Then they do not come apart until the tRNA will signal, or the lack of tRNA will signal for disassembly of that. So once it starts going, it just keeps on going. You still have a lot of more energy that is going to drive this synthesis of polypeptide on the way. But then in order to get this together, you needed that initial amount of energy to assemble. This is the overall picture. You can study in a whole compacted form. Okay. And that is just another showing of what this strand organelle sequence looks like. Of course, you don't have to memorize that, but you have to know here there is a signal that will say to the ribosomal, small ribosomal subunit of prokaryotes that the start codon is around the corner. Okay, so this is signaling to the ribosome that protein synthesis is going to start soon.